In this particular image, we'll take a look at the replication strategy for plus-stranded RNA viruses. In the case of plus-stranded RNA viruses, you'll see that once the plus strand is introduced, we're going to actually have the direct template necessary in order to enter translation. Recall from the previous DNA virus video that you need plus-stranded RNA in order to translate that into a viral protein. So since what entered the cell was already a plus strand, that can proceed directly into translation and allow for the production of viral proteins. Once you begin to produce viral proteins, some of those viral proteins that are produced will be a replicase. The replicase then can go and lead to the production of additional viral genome. Ultimately, that replicase will produce minus-stranded RNA, and then once you have the minus-stranded RNA, the replicase can use that as a template to make more plus-stranded RNA. The plus-stranded RNA that's produced can either be incorporated into new variants, or it can be used as a template to produce more viral proteins. Now you might be wondering, why doesn't replicase just make plus-stranded RNA from plus-stranded RNA? Well, it goes back to what I told you in the previous video regarding DNA viruses. Essentially, with nucleic acids, you can never go from a plus strand to a plus strand. You have to go from a plus strand to a minus strand. In other words, you always make the complementary strand. So in this case, even though we're starting with plus-stranded RNA, we can't directly make more plus-stranded RNA. That's because this plus-stranded RNA can only serve as a template to make the minus strand. Once the replicase produces a minus strand, then it has the new template in the minus-stranded RNA in order to convert that or synthesize the plus-stranded RNA molecule. So even here with the RNA viruses, we can't make a plus strand from a plus strand. You still must always go plus strand to minus strand, and minus strand ultimately to the plus strand. So once again here, with a single-stranded RNA virus that has a plus strand, once that plus-stranded RNA enters the host cell, it can proceed directly into translation. It's already a plus strand of RNA. That plus strand can be translated into additional viral proteins, one of which may be a replicase. The replicase can then take the plus-stranded RNA, use that to synthesize the complementary strand, which will be the minus strand of the RNA, and then that minus strand can serve as our template to make more additional viral genome of a positive stranded RNA molecule. Now, let's talk about minus-stranded RNA viruses. As you can see, this will be obviously different. The minus strand cannot directly enter translation. So, what happens is that minus-stranded RNA virus must also carry with it its replicase. So when you look at minus-stranded RNA viruses, the viral genome along with the replicase are both contained within the capsid. Once the genome and the replicase are released, the replicase can then use that minus strand of RNA as a template to produce more positive strand RNA. And now once we've got our positive strand or plus strand RNA molecule, we can proceed and go ahead and carry out translation. The plus stranded RNA molecule can also serve as a template for the replicase in order to produce additional minus stranded RNA molecules. So now we'd have our viral proteins along with our viral genome and the ability to make more virion. Next up, double-stranded RNA. Remember, with double-stranded RNA, you're going to have both the plus and the minus strand. Once again, you're going to have both the viral genome and the replicase within the nucleic capsid. Once the viral genome along with the replicase enter the host cell, you're going to be able to unzip this double-stranded RNA molecule, and then you can use the plus strand of that RNA molecule, the minus strand of the RNA molecule as a template for the synthesis of plus-stranded RNA. Once you have your plus-stranded RNA again, you can feed that into the process of translation in order to produce more viral proteins. Now, we run into another issue because, well, we need double-stranded RNA in this situation. So in that case, the replicase can use the plus strand as a template to synthesize the minus. And once you've got minus stranded RNA, you can allow the two to come together and form a double strand of RNA. So essentially, your major component that's really critical here within a double-stranded RNA molecule, or a double-stranded RNA virus, would be the minus strand of the RNA. The minus strand of that double-stranded molecule is used to synthesize the plus strand RNA in a free form. The plus strand that came in with the original viral genome would be bound to the minus strand. That wouldn't help us at all. So once we have our free plus stranded RNA, we can translate that to a protein. We can also use it as a template in order to make our minus strand, and then combine the two again in order to make our viral genome. Now, we want to talk about some issues that can arise with these RNA viruses. With RNA viruses, it is possible for some issues to arise, and these actually play a critical role in the lives of humans. RNA viruses, once again, depend on a replicase. The replicase is necessary because, well, as humans, we don't have the ability to make RNA from RNA. Well, the replicase, unlike the enzymes that replicate DNA, the replicase that produces RNA from RNA does not proofread. So ultimately what that means is that there's a higher frequency of mistakes in replicating viral genome. This leads to two potential effects that are seen in RNA viruses. The first is called antigenic drift. When the replicase makes random mistakes, these mistakes can lead to mutations. And these mutations can change the surface proteins and the antigens present within a viral envelope or within the surface of a naked cell if it doesn't have an envelope. At the end of the day, these mutations can make it more difficult for our immune systems to combat those viruses. This is called antigenic drift because, well, essentially, the mutation may cause a shift in the reading of that RNA molecule, and it, well, for lack of a better way of putting it, drifts to the next codon. Now, if you look at some of the RNA viruses, they have a segmented genome. Segmented genomes are those that can be separated and, well, they're combined, and as long as you have all the segments, well, you can get a complete virion. In the case where two RNA viruses infect the same host cell, you can actually have two completely different viral genomes. I love the analogy that your book actually gives for this. They represent two segmented genomes by using letters A through E. The first virus, they let that one have a viral genome of capital A, capital B, capital C, capital D, and capital E. Each individual letter is a segment of the complete genome. They let the second virus have a genome represented by lowercase letters. Lowercase a, lowercase b, lowercase c, lowercase d, and lowercase e. Now, as long as the resulting virion has one A, one B, one C, one D, and one E segment, it would be considered a complete virion. Well, the issue comes in is because both of those viruses infect the same host cell, you've got all the different possibilities. You've got all the capital letters and all the lowercase. So say if the original virus once again had capital A, capital B, capital C, capital D, and capital E, well, because the presence of the lowercase versions of those segments was there, maybe the resulting virion has capital A, capital B, but then lowercase c, d, and e. That would be a completely new virion. We call those new subtypes. And these new subtypes ultimately become, or rather lead to, what we call antigenic shift. The shift is when you completely move things around. Not just you drift from side to side, you literally are moving segments of the genome and, well, for lack of a better way of putting it, shifting them around. This is one of the reasons why the influenza virus is so problematic. The influenza virus carries out antigenic shift repeatedly. The influenza virus has a segmented genome, and as it shifts around those various segments, it eventually allows the virus, that is, to evade our immune system. Next here, we have replication of reverse transcribing viruses. Reverse transcribing viruses go in the complete opposite direction of what we're used to seeing. And that would be, instead of making DNA, or rather, instead of being able to make RNA from DNA, the reverse transcribing viruses actually make DNA from RNA. Completely different process. It's the exact opposite of what we're used to seeing. In order to do so, these reverse transcribing viruses carry with them a reverse transcriptase. As you can see, the reverse transcriptase molecule makes DNA from RNA. So we'll start with our plus-stranded RNA. Yes, you can send this directly into protein synthesis, but that's not what happens. The reverse transcriptase allows for us to produce a minus strand of DNA from our plus strand of RNA. Once you've got your minus strand of DNA, that can actually move into, well, transcription. You can utilize the minus strand of DNA to make a plus strand. The plus stranded DNA then combines with the minus strand, and you'll have double-stranded DNA. And from here, things get somewhat interesting. Typically, once you've got that double-stranded DNA molecule, it will integrate itself into the host cell's chromosome. And that integration allows for it to become somewhat of a silent genome. In other words, that host cell could replicate the DNA over and over again, and as it continues to replicate it, well, ultimately, you will continue to see that host cell promote or pass on the viral genome to future cells or progeny cells. At some point in the future, that double-stranded DNA that was incorporated can be excised, and once excised, the minus strand that's carried in that double-stranded DNA could serve as a template for transcription to produce the plus-stranded RNA. Plus-stranded RNA feeds into translation, and the minus strand would also allow you to ultimately transcribe more plus-stranded RNA. The benefit of this is that these viruses incorporate into the host cell's genome. Therefore, they can continue to propagate because they are literally within the host cell's genome instead of being outside of it, which is in some cases a target for our immune system. Viruses such as HIV and also hepatitis B are going to be examples of reverse transcribing viruses.